Good morning, my name is Chady Nasrallah. I'm a research scientist here at the Oxford site at Vertex.
I'm Rachel Hossack. I'm a research scientist at Vertex at our Oxford site. And I'm arriving at work for a day of science.
You are focusing on a disease that is quite challenging. There is no treatment at all for it.
My job is trying to make the protein, understand the structure. Then we can try and make a molecule that can correct this protein and treat the disease so that these patients can have a better quality of life. I'm going to meet up with Chady to talk about construct design for the proteins that we want to try and purify this month and all of that cool stuff. We started off the day looking at construct designs for some really challenging proteins that we're struggling to express and purify at the moment.
We try to use technologies that will bring us a very specific details within the protein structure that will enable us to improve properties of this protein. We can try maybe a couple of variations.
We designed some new constructs that will help not only the expression in the cells, but help us to be able to pull out purer protein. We'll get some of those constructs ordered, do the test expressions, and then put them into some cells.
Sounds good. Exciting, yeah.
Yeah, I think this one's going to work.
Yeah, thanks, Rachel.
So we've designed the DNA construct, and in this lab, we add the DNA to the cells and use the cell's protein production machinery to make our protein of interest. So I've prepared about 12 liters of cells. So I need to just take a small 1/2 ml sample of my 3 liters of cells, and that will go onto the cell counter machine. I'll do a cell count on the cells to see if they're healthy. If they are, in my biological safety cabinet, I'll add the DNA, which Chady and I originally designed, and add that into the cells. Leave it a couple of days for the host cell machinery to make the protein. After 2 days or so, harvest the cells and then take those cells forward to the purification process. So I'm at the final step of the purification of the protein. Before I hand over to Chady, I need the protein as pure as possible so that he can get a really good cryo-EM structure. So I've got my protein, which is partially purified, and now I'm going to inject it into the actor, which will separate all the proteins in the solution on the basis of size. And we've got about half an hour while it runs. I'm going to load a small sample of my protein into this gel. You add an electrical current and that will move the protein down through the gel. The gel will separate the proteins in that particular solution, again on the basis of size, so I can determine the purity of my protein. So I've got my final protein, which is 100% pure, ready for grid, so I'm going to hand it over to Chady now.
And now I'll be performing the final quality control step using the Nano DSF. I just received the purified protein from Rachel, and we will try to measure the stability of the protein. So this contains my protein, and I'm gonna start by prepping the condition for Nano DSF. I'm gonna pipe this protein into different buffer conditions. This is basically a mix of pH and salt. We want to find out where the protein is happy in that condition. Once we have the protein here, we're gonna just do a brief spin using this centrifuge here to get rid of any air bubble inside the wells. So now we are next to the Nano DSF machine to measure stability. We will heat up the protein sample, and the protein sample at some stage is going to unfold and will give us a TM value which reflects the stability of the protein. So if we find a condition where the protein stability is higher, this will be something that we will move ahead with and test in cryo-EM. Now we have the protein fully characterized. The first step is to freeze the protein in a very thin layer of ice, and we will be able to image them using cryo-EM. And then I'm going to take 1 by 1 the grids, deposit it on this cover slip. Starting here is a plasma cleaner to get it ready for the freezing step. There's actually an equipment important to do the plunge freezing, and we will try to freeze the protein in, again, cryogenic condition. And the last bit would be actually preparing the grids for screening for the microscope. And we're gonna use this apparatus here that will let us basically do what we call clipping the grids to be ready for the screening. Once the sample are shipped in this container, we're gonna analyze the data and then review it at a high resolution. And we will communicate back this information to our medicinal chemists for them to improve a bit more the design of this small molecule and hopefully improving patient life in general.